This lesson is about transport across the membrane. So we have quite a lot to cover here, but it is based on our describe the structure and function, and then we expand from that. So we've discussed the structure of the cell membrane, and we can now use this knowledge to learn how this membrane is selectively permeable. So it's allowing different substances to come and go when required. For a cell to function and perform all those basic requirements for life, it's got to gain those nutrients from its surrounds. So it is selectively permeable, and it depends what it needs as to what and how a substance actually enters and exits the cell. There are two types of membrane transport. One is passive, it just kind of happens. It's based on the concentration gradient, and it does not require any energy to happen. And then there is active transport, which actually requires energy, and our energy in our body is ATP. That's our cell currency, our cell energy currency. So we have three types here of passive. We have osmosis, diffusion, facilitated diffusion, and then ion pumps, endo and exocytosis. They're the ones we'll be focusing on. But we have to be clear on what a concentration gradient actually is before we can understand how membrane transport works, because it's going to impact both active and passive. So particles will distribute themselves across a space from really high concentration, so obviously at this end, to really low concentration. Essentially, they just want to move somewhere that is slightly less packed, right? It makes sense. So they'll travel from an area of high concentration to low concentration and spread themselves out. So they are mostly even. They want to evenly distribute. So let's start with passive movement. All right, firstly, let's talk about diffusion. It is the spreading out of particles from an area of high concentration to low concentration. So it's working with the concentration gradient. So say we're standing in the corner of the sports hall, someone sprays Lynx deodorant. Eventually all those particles are going to diffuse out across the entire sports hall so that unfortunately everybody can smell them. We can see that happening in this little animation. Some things that might impact how quickly the process of diffusion actually happens will include temperature. So the hotter it is, the faster those particles are going to move, so vibrate, and then the faster they'll move across the membrane. The steepness of the gradient, so if there are way more particles on one side, they're going to move quickly to evenly distribute themselves, and the size of the molecules actually doing the diffusion. Now this is a really nice animation, but unfortunately it's a little bit inaccurate. Yes, things are going to move from one side to the other to evenly spread out, but they don't just stop there. They will continue to move back and forth in that dynamic equilibrium. Facilitated diffusion is just that. It is being helped by something, but it is still considered to be diffusion, right? The center of the membrane is really hydrophobic. So positive and negative charged ions in solution are going to struggle to get through. So protein channels can help them out. We know that there's proteins embedded in that cell membrane. So each cell is going to control what type of channels they make for their membranes to control what comes and goes. But remember, this is still a passive process. It's just working with the concentration gradient. Osmosis is the movement of water across the concentration gradient. So essentially it's just diffusion of water, right? Water as a solvent, so other things are dissolving into it. It's more mobile than the solute particles, which need those channels to facilitate movement in and out of the cell. So water is trying in a dynamic way to even out the concentrations where it can as well. So it's trying to help out. Water can pass between the phospholipids in that cell membrane, but they're also aquaporins, and these are protein channels used for bulk movement as well. So they're kind of moving single file through here, and the internal parts of the protein channel are hydrophilic, so they like the charged water particles, whereas the phospholipids don't. All right, let's talk about active movement. Now in the cell membrane, some of the transmembrane proteins are carrier proteins, as opposed to say those channel proteins where things can just move really quickly through them. And these channels are transporting substances against concentration gradient, and that's what makes it active movement across that membrane. There needs to be energy expended. So ions are entering, proteins change the shape to allow the ions to exit on the other side. It's kind of like an elevator where the doors open both sides. You walk in one side, but you come out the other. And obviously this process is much slower, but very, very specifically. Importantly, we are moving substances against that concentration gradient. I'm going to keep repeating it. We have to spend some energy to do this type of transport. With active transport proteins, there are, plenty of different examples in the cell. Axons in nerve cells will do this with sodium and potassium to create an actual nerve impulse. They're very complex and very efficient proteins in that cell. Endocytosis is used to transport really large things that may not actually fit through those protein channels. So we're talking antibodies, food for digestion, bacteria to destroy, whatever it is. It's the fluidity of the membrane that actually allows for this to happen. And the membrane opens up, restructures itself, and then closes itself back up, which is pretty spectacular. So this is the process known as phagocytosis, which is cell eating, which is a type of endocytosis, which is exactly what these particular white blood cells are doing, trying to chase down a bacteria, and then will eventually endocytose it and bring it inside of its cell membrane. Exocytosis is essentially the same process, but in reverse. So vesicles, those little bubbles, are made by the phospholipid bilayer. It's fusing with the external membrane to open up and release proteins, enzymes, whatever it is. So this here is the outside of the cell. Now, digestive enzymes or proteins are made in the ribosomes and the ER. They go to the Golgi body and then they are put into one of these vesicles and they are expelled. So we can actually remove excess water like this, and paramecium, so single cell organisms, have these little contractile vacuoles that can do that with water as well. Now, Oxford has a really lovely table showing some of the features of active and passive transport as a comparison. That's our starting point. But firstly, let's make sure we understand what the QCAA is actually asking for when they ask us to compare something, because we are aiming to compare active and passive transport. So this is the definition of compare from QCAA. It says display recognition of similarities and differences, that's the first part, and recognize the significance of these differences and similarities. Okay, so there's three things there that you need to do. And basically they should be prompting you if they want those significances now. They've changed the way they do things, but basically we still need to follow this. So always think about those three things, and we'll go through this in class, but it's worth trying it out yourself with the knowledge that you have to try and find similarities, differences, and the significance. So what makes these things actually important? Why does it matter that there are these similarities and these differences? So remembering that we've got a lot of dot points here and we are building on that, describe the structure and function to understand how to explain the movement of particles or substances in and out of the cell membrane.